So many people are talking about longevity online, but few are actually at the frontiers of research. Today, we're going to speak with Dr. Jennifer Garrison, a scientist from the Buck Institute for Research on Aging, whose research has the ability to impact the future of medicine and how we understand aging as a species. We're traveling to San Francisco to meet with Dr. Garrison in her home, where we'll get into big questions like, what do we currently understand about aging? And is there evidence to suggest we can live forever? We'll learn about female reproductive aging and how it may actually hold the key to longer health spans for all humans. Quick little heads up, this is my first live interview and we ran into some technical issues, especially with the audio. I'm working on smoothing these out for future interviews, but I promise the insights in this conversation are worth the listen. So thanks for bearing with me on this one, and thank you, Dr. Garrison, for taking a chance on a woman with a camera and a whole lot of questions. Let's get started.
I was more interested in normal aging, what's happening in someone who doesn't have a major disease. Why is it that there's this stereotyped decline across all of our organ systems? It happens at different rates in different people, but you know approximately how old someone is by looking at them. We have to start thinking about all of women's health focused through the lens of ovarian function. I've stopped calling them reproductive organs. Calling them reproductive organs really
narrows our lens.
It narrows our lens. That's a good way to put it. Fertility is very important. It's not the whole story. And I think it has allowed people to make women's health small. My name is Jennifer Garrison. I am a faculty member at the Buck Institute for Research on Aging. I also have appointments at UCSF and at USC. I am the executive director and, this is a mouthful. I'm the executive director and co-founder of productivehealth.org, which was formerly called the Global Consortium for Reproductive Longevity and Equality. But as you're going to see in this conversation, we have to stop calling them reproductive organs. So I can't have reproductive in the name of this organization.
What is your bigger why of why you do the research that you do?
I do what I do because it's worth my time. So I spend a lot of time thinking about how I can make the most impact doing the research that we do in my lab. If at any point I ever think that what we're doing isn't really pushing things forward, or if I think that somebody else can do what we do, then I'll stop that. But for now, I think what we're doing is really important. I spend a lot of my time doing things that aren't really my academic job. I started this center and global consortium to try to push forward research around female aging. And I think that that is maybe the most important thing I could possibly do. A few years ago, many years ago now, I came to this place where I realized I knew nothing about how my body works. I'm a scientist. I'm a curious scientist, I think. And there was a moment where I was kind of forced to confront the fact that I had never really thought too much about how my body works, how my ovaries work, just curious, wanting to know more, a little bit embarrassed that I didn't know more. I realized that there's not that much to know. Right? I'm sure you're female, so I imagine that you've had similar experiences, right? For whatever reason. And then if you go down the rabbit hole, you start to understand why there's such a huge data gap. For me, at least, was abject rage, really a lot of anger. And then, what can I personally do to make this hopefully better?
I'm gonna zoom out for a moment because a lot of the things that you're touching upon are related to, I would say, one of your expertise, which is neuropeptides and how they change in aging. So can you introduce us to the concept of a neuropeptide for people watching who may not really understand what that is and why it's so important?
Yeah, sure. I should say neuropeptide is a misnomer. I didn't tell you that ahead of time because I think it's an important thing to just point out in real time. I tend to call them bioactive peptides now because they're called neuropeptides. It's like a historic relic because they were discovered in the brain and the nervous system, but they can be made and sensed by non-neuronal cells. And so it's kind of silly to call them neuropeptides. Not all of them are in the nervous system.
Quick question to clarify there. When you say bioactive peptide, does that mean it can also be made by other organs, or these are, you're still focused?
Yeah.
Okay, so these are any peptides that are
any endogenous peptide.
endogenous peptides.
Not something that you're introducing from outside.
Okay, so essentially the peptide hormones is what you're studying?
Well, there's a lot of nuance here in language, and words do matter, I think. And your public should actually understand this, especially if they care about this kind of signaling. And that's true for men and women. But the word hormone, what does that word mean to you?
To me, it means a signaling molecule that is released through the bloodstream and acts on other organs in the body than perhaps where it originated. And an autocrine would be acting on its own tissue.
Okay, yeah, I think that's reasonable. Maybe a slightly broader view would be any signaling molecule that carries a message between two cells that are not connected physically. And so they could be, in the brain, a millimeter is a huge distance, right? It could be that they're signaling across what we think of as very small distances, but if they're signaling between two cells or two groups of cells, could be tissue, but could just be groups of cells that are not physically connected, that's a hormone. I talk about bioactive peptides as like the brain's Wi-Fi. Hormone is like this umbrella term. Bioactive peptides fit under that. And some bioactive peptides are not hormones. They signal only at synapses, right? So I try to break it down into wired signaling, right? So the connectome, synapses, anatomical wiring, which is literally like a physical connection between two neurons, right? Neuropeptides can signal synaptically in response to an action potential, but they can also be released from other parts of the cell. They can be released from non-neuronal cells, and they can signal over long distances as well. And so there's like the wired signaling and then the wireless signaling, or the Wi-Fi. So they do both. And hormones, are more on the Wi-Fi side, but hormone could mean a small molecule, it could mean a lipid, it could mean a gas, it could mean a bioactive peptide. It's not related to just bioactive peptides.
I'm glad you corrected me that it's actually bioactive peptides. What do we understand in how they regulate our very basic homeostatic functions in our body?
Yeah, so if you're talking about homeostasis, you're really talking about energy and fluid balance, body temperature regulation, reproductive function, and circadian rhythms. Those are all mediated by bioactive peptides in part, right? There's always going to be synaptic signaling when you're talking about the brain, but in part for the more long-term aspects of those particular systems. And maybe it's easier to give an example. So something like there are systems in your kidney that are there to conserve water. So, what will happen when you're thirsty is that there'll be vasopressin, which is a bioactive peptide, will go to your kidney and basically promote fluid reuptake so that you're keeping as much fluid as possible and not getting rid of fluid through urinating. That's just one example. When I think about brain function, you can break it down in a lot of different ways, but one way to break it down is that you have the anatomy, like the physical connections between the cells, and that is what we would call the connectome. So that's like the map of all the synapses and all the gap junctions. And then you have activity. So you said electrical activity. So there's electrical activity, which can specify active circuits. And electrical activity can also change where synapses are and how strong they are. In a slightly overlapping way, then you have flexible modification. And so I think of bioactive peptides as like encoding flexibility in the nervous system. Does that make sense? You can think about it actually, if you want to carry forward the analogy of the anatomic connectome, which is absolutely required if we're going to understand nervous system function, but it doesn't explain everything, right? We've had the connectome for several animals. For C. elegans, we've had the connectome for 35 plus years. And we still can't, in this organism where we've mapped out every neuron has a name, there's only 302 of them. We know all the 8,000 synapses, all the 800 gap junctions. Like we literally have the roadmap, but we can't predict how information will flow through a circuit to mediate something like a behavior because there's the anatomic connectome, then there's this like second connectome of biochemicals, neuromodulators, of which neuropeptides are one class, that can essentially change the functional output of a circuit without changing the anatomy. And so what we need to do is understand this biochemical connectome and then what's the transform between the two, or the relationship between the two. For neuropeptides, the reason that they're so interesting is because they're mediating not just the physiology. So like for that fluid reuptake in your kidney, your hypothalamus senses that you have some imbalance in triggers you to be thirsty and drive you to drink. That's a behavior. And then it's also mediating the physiology, which is the fluid reuptake by your kidney, right? It's super cool that they can do these two very different things.
I have so many questions related to this. Your research is part of the Buck Institute, which is studying aging and longevity. How does this relation between these bioactive peptides and our behavior and the associated physiology, why is this so relevant for aging?
I mean, this is, to be clear, this is my research program, and so I think it's very interesting. I don't know how much other people think it's interesting, but for me, when I started my lab, I trained as a chemist, so I have a PhD in chemistry and chemical biology from UCSF. Then I did a postdoc in New York with Corey Bargman at the Rockefeller University in neuroscience, studying neural circuits and behavior. When I started my own lab, I hadn't worked on aging before. It was new to me. So what I'm interested in is really just like a mix of all of the things that I think are cool in science applied to aging. And so when I started thinking about aging, trying to consider what are the most upstream intervention points that we can think about. Like, what's the first domino to fall with respect to, in this case, systemic aging? Certainly when peptidergic systems get dysregulated, that can cause all kinds of different diseases, and we can talk about that. Not surprisingly, when you consider what they're doing, right? But I was more interested in like normal aging. Like, what's happening in someone who doesn't have a major disease, what's happening in their bodies during aging? Why is it that there's this stereotyped decline across all of our organ systems? It happens at different rates in different people, but you know approximately how old someone is by looking at them. So if we're thinking about like what's the first domino to fall with respect to systemic aging and normal aging, what do you think about when you think about somebody aging?
Well, I'm maybe primed a little bit by doing research for this conversation, and it's, I think, very relevant to me. But for example, not just the physical aging on our outside, our skin. I'm now in my 30s. I'm already noticing my skin looks a little bit different, but I'm also thinking about my own reproductive system because I don't have children, and I know that there's a biological clock that's ticking. And it was so fascinating to research for this conversation and realize that is one of the first points in decline in the human body, is the sudden nosedive that the female reproductive system takes. I mean, it's not yet there for me, but at menopause, which is now going to be midlife as we live longer, I'm really thinking of that as a first aging moment.
Okay, totally. Yeah. Skin, female reproductive system, changes in energy and metabolism are really common, changes in sleep.
Also think of sarcopenia,
Sarcopenia, muscle loss. Yeah. And so a lot of these things really do come back to those homeostatic systems in the hypothalamus. And we know that there is, and this is not my work, but we know that there's an age-related increase in inflammation in the hypothalamus. There's an increase in inflammation everywhere in your body with age, but it's not, in the brain, it's not uniform. It's not happening to the same extent everywhere. In the hypothalamus, it seems really to be a hot spot. And so, like, very naively, the idea going into what I work on now was, okay, there's all these really important circuits in the hypothalamus. Many of them are like really neurochemically diverse, lots of neuropeptides, lots of things happening there. They're mediating these incredibly important homeostatic systems. So what if, as you age and there's this age-related increase in inflammation that disrupts some of those circuits, in that way disrupts the Wi-Fi signaling that these bioactive peptides are mediating to control these systems. Compared to what we know about synaptic signaling, which is a pretty, we understand in quite a lot of detail how that works, thanks to decades of research from dozens, if not hundreds of labs doing this beautiful work, which was made possible because they are physically connected. So you can record from the presynaptic neuron and the postsynaptic neuron and know exactly what's happening there and really study that process. Whereas with bioactive peptides, even if it's a small distance, if they're released from one place and then they're binding to a receptor in another place, they're signaling over maybe long distances, maybe long time scales. How do you study that? Like, how do you measure that? And then it gets even more complicated because one neuropeptide or one bioactive peptide, you were trying to get at this at the beginning of our conversation, can be made by lots of different tissues, maybe, and one peptide might have multiple receptors that are expressed in different places. And so it becomes this combinatorial nightmare. And then layer onto that, there's hundreds of peptides, hundreds of receptors. One receptor might sense multiple peptides. One peptide might bind to multiple receptors. If a neuron or a cell is making a neuropeptide, so like there's orexin neurons in the hypothalamus, they're almost certainly making a dozen peptides. So it's rare that you see a cell that's making just one peptide. To me, that says, well, maybe there's some combinatorial code. Like maybe it's more important what cocktail a cell makes than any individual peptide. But all the research that has been done, for the most part, has been pick your favorite peptide and study the heck out of it. GLP-1, oxytocin. Yeah, just, and study the heck out of it in isolation, partly because there weren't a lot of, the tools just weren't there, but also, because our thinking has evolved, our knowledge has expanded, and now we're looking at the nervous system in a different way and taking into account all this complexity. So we're just kind of at the beginning of learning how this stuff works. So the real question, and it sounds very basic, but the real question is, how do neuropeptides change with age? Like full stop. In whatever tissue or biofluid you want to study it, how do they change? Like as a class, not just one at a time, but as a group. This even gets more complicated. I'm just going to keep going down a rabbit hole if you don't stop me.
I love it. Go down the rabbit hole and then we'll bring it back.
Okay.
Because I'm also very fascinated by this.
All right. Aside from the challenge with studying something that's long time scales and long distances, is we haven't really had a good way to get a handle on, there's no like proxy for measuring neuropeptides. You can look at mRNA, you can do sequencing, you can do RNA-seq and all these kinds of beautiful omics that really belong at the level of RNA and DNA. But unfortunately, those don't reflect the levels of active peptide, because between DNA, RNA, and then the actual peptide, what happens is they're made the same way that proteins are made. So go DNA, RNA, protein, but in this case, it's a pro-protein precursor where it's a long chain of amino acids where the peptide is just one little piece of it. And it gets packaged into a vesicle that it will eventually be released from. And then there's a bunch of enzymes in there that cut it out and modify it and decorate it and make it into this relatively stable structure that once it's released will go do its signaling. But that active peptide gets stored in these vesicles and, it can sit there for a long time. And so the mRNA it tells you that you can make that peptide in that cell, but it doesn't tell you anything about the levels of peptide or when it's being released or when it's signaling. And so you really have to measure bioactive peptides by mass spec, which is challenging. And so there's just all these little things that have made it so hard to get a handle on what's happening that this is where most of the first 5 years of my lab were, is just tool building, just build tools.
So to zoom out, you have these bioactive peptides, which in and of themselves are very hard to measure. You have this web of different peptides coming from the same tissue, responding to multiple receptors, and you're thinking that also it's more likely that these peptides are also working in these kind of cocktails. And that just sounds like a lot of information to process, especially when there's the kind of the next level of what does this mean in terms of medicine and therapeutics. So how are you starting to untangle this crazy web, and where do you see this being relevant and powerful for us as humans and medically?
Yeah. Well, luckily for us, this is a signaling system that evolved early. So there's a lot of good model systems to study this in. And also, luckily for us, they're signaling through GPCRs, G protein coupled receptors, not through ionotropic receptors like classical neurotransmitters. And so that means two things. It means that there's actually a lot of pharmacology, decades of pharmacology around GPCRs. Those are very druggable. We call them druggable targets. And so for oxytocin and vasopressin, just to carry that example through, we've known for a long time, those were the very first bioactive peptides that were discovered, that were characterized, and that were synthesized, right? So, Nobel Prizes were won for isolating oxytocin and vasopressin from, doesn't matter, but it's a cool history. And then for figuring out what they were, like what they actually are. Do you know what they are?
Strings of amino acids?
Yeah, but do you know anything about how big they are?
I don't. Can I guess?
Yeah, guess.
29 amino acids.
Okay, that's like fair. A protein might be somewhere between 1 and 300, right? Or something like that.
So I imagine
it's a peptide.
not too long.
So we talked a little bit about vasopressin. We didn't talk about what oxytocin does. And the reason we talk about oxytocin and vasopressin in the same breath is because it arose from a gene duplication event. So they're actually almost identical. Oxytocin and vasopressin are 9 amino acids, only 9.
Crazy.
And they're different only at 2 positions. And then they have multiple different modifications that essentially make them very rigid and very resistant to non-specific degradation, right? Because they're released from a cell and they have to go somewhere to do something. And so you don't want them to get chewed up by non-specific proteases or peptidases. So even though they're small, they're quite mighty. And the way that they work differently is they have different receptors. But there's also the opportunity for oxytocin to bind to vasopressin receptors and for vasopressin to bind to oxytocin receptors. And so this is like emblematic of how peptides work. There's often redundancy. So in this case, there's a little redundancy here. Vasopressin can almost entirely substitute for oxytocin. Oxytocin cannot fully substitute for vasopressin, partially can, but that's why there's no true genetic knockout of vasopressin because those animals are dead. But there are knockouts of oxytocin in mice that have almost no phenotype, which is crazy when you think about what we already know the peptide does. And it's because vasopressin can really like substitute for it.
Wow. So I'm hearing there's hundreds of bioactive peptides, but still if you think of how many different things physiologically, behaviorally, these bioactive peptides are regulating, it's in the redundancy, it's in the cocktails, it's in these crazy combinations where you get so many more, perhaps, possibilities from this very small select group of bioactive peptides. Would you say that's correct?
I think that's about right. Yeah, okay. Simple, right? It's like no big deal.
Why this in terms of aging and why this in terms of aging in women? Because I'd love to spend some time.
We'll get there, sure.
Yeah, I'm wanting more conversations around aging in females, so.
Okay, I'm your girl. I'm here for that. For me, like I said, when I think about systemic aging, it really is those homeostats that change. And I think, the hypothalamus, those circuits are rich with peptidergic signaling. It's a kind of a black box. We know something about what the peptide does, often because of like genetic experiments, but then how they change with age. And there are some studies that just measure endpoints, and some peptides go down, some go up. I think there's just a huge opportunity to think through what's happening. And yes, we do have to measure things and generate hypotheses based on what we see. But I think it's a really rich and likely important node of, I don't want to say regulation because that makes it sound like it's a program, but a node of potential for intervention that is relatively straightforward once you know what's changing and how, and you can, very testable hypotheses, and also relatively easy and straightforward ways to drug these systems. So for me, it's kind of a no-brainer. And honestly, I hadn't thought about the female reproductive system until about 6 years ago. When we were studying oxytocin, we discovered that it definitely does play a role in aging, and trying to figure out how it was working. Right? So you get to this okay, it's playing a role in aging. How is it doing that? It turned out to be working through the reproductive system. And so we sort of accidentally backed into thinking about the reproductive system. And that was right at the moment when Buck started a center focused on healthy aging in women and focused on thinking about aging in the female reproductive tract as a lens through which to potentially extend health span. I know that sounds kind of like wordy and a little bit.
No, it's amazing because I think this is also for any men watching, maybe besides caring about the females in their life, they can finally get behind studying the female reproductive system.
Oh, sure. Yeah. I mean, I give talks all the time to men, and there's a few things that make them wake up from looking at their phones. Actually, usually on the title slide, there's usually something about females on my title slide. And so that automatically means that half the audience is checked out until I say, women live longer than men, right? On average, yeah, they live longer than men, but they spend a significantly longer period of time in poor health if they're lucky enough to live to old age. So we're talking, I think for this conversation, we're talking about women and men who live to old age. That would be 70s, 80s, 90s, and beyond. For those people, if you're female, you are going to spend about 25% of your life longer than men in that period of time at the end of life when you are afflicted by multiple age-related disorders. If you turn it around, that means that females have a shorter health span than males. That's crazy. Why is that? It's directly because of aging in ovaries. Directly, because your ovaries are aging faster than the rest of the tissues in your body. And that shortens your health span.
And so when a woman enters perimenopause, their health span is immediately going to start?
No, it's not even that. Menopause is a hallmark of ovarian aging, right? It's a very measurable hallmark, but it's not, I would say, I'm trying hard now not to bookend this conversation with just fertility and menopause. And that it's an evolution of my thinking, because I definitely did frame the conversation that way 4 years ago. And those two things are very important, and also measurable moments where you can measure ovarian aging in a person.
Well, as I mentioned, I'm now 32. I am cognitively aware that my ovaries have been aging and they're already on a decline, which is wild because I'm not yet thinking about menopause.
But how do you know that? Have you measured something that tells you that? Because I think at the level of the individual,
it can be very variable.
Very variable.
So on average.
On average,
at
yes.
My age, it's starting to decline.
Okay, fair.
But I have not. I would love to do some extensive. I don't know if these tests exist, but
I wish they did.
Maybe they will in the future. But it's something that I'm already thinking about, even though, menopause is not on my mind. The aging of my ovaries and the quality of my egg cells, as someone who does want to conceive at some point, that is very much on my mind. And I know, being a millennial, only one of my really good friends has a child. The rest, all the women in my circle, are not having children, and they're still thinking about, do I want children? Do I want to freeze my eggs? This is very top of mind, even for younger women who are not yet in middle age.
Story old as time. Yeah. I mean, really, my generation was there too. I'm a little older than you, though. We're past that now. But I think maybe a more expansive and a richer and more accurate way to think about it is ovaries are endocrine organs, right? So and that really just means that they make hormones. They make long-range signaling molecules that mediate conversations with distant tissues. And we think a lot about brain-ovary signaling and that chemical conversation that goes in both directions. But if you think about ovaries as endocrine organs, right, separately from fertility, we know very little about how ovaries work. But what we do know is what happens when you take them away. Or when they're not working properly, right? So we infer everything from those kinds of experiments and real life examples. So if ovaries are sitting at the center of a signaling network in a female body, where ovaries talk to your brain for sure, but also your heart and your bone and your liver and your skin and your muscle and almost every tissue in your body. And every single one of those axes of communication is unique and complex, and I don't want to make it sound trivial, because it's not. And we don't understand many of them, but they're there. And we know that because we know what happens when you take ovaries away or when they're not working. And so if that's true, then I think what we can infer is that ovaries are promoting health. That's what they're doing. And so when they're not working properly or when they're absent, that uncovers different health risks. And the health risks that are uncovered are dependent on how old you are, what your genetic risk factors are, and what maybe your environmental exposures have been, so who you are as an individual. When I think about it now, it really applies to women at any age. For girls who haven't gone through puberty yet, if ovaries aren't working properly, that could uncover risk for having issues going through puberty or having irregular menstrual cycles or something like that. And then for women in their 20s, if ovaries aren't working properly, that can uncover risk for things like polycystic ovarian syndrome, PCOS, or POI.
Increased osteoporosis risk later in life, perhaps?
Definitely. So if you just step through your lifespan, so you're in your 30s. So if ovaries aren't working properly, that could mean that you are going to encounter infertility or have miscarriages, potentially have a increased cancer risk. if ovaries aren't working properly, or maybe a better way to say it is when ovaries start to fluctuate in function because they go up and down at perimenopause, then that uncovers risk for all of those health issues that are associated with the 135 symptoms of perimenopause and counting, right? And then at and after menopause, it uncovers all those risks that I think we've talked about a lot in the last few years, things like increased risk for cardiovascular disease, stroke, osteoporosis, sarcopenia, autoimmune disorders. I can keep going, cognitive decline, neurodegenerative disease. And so if you buy that, like I'm telling you something that's like, this is how I think about things. If you buy what I just said, then we have to start thinking about all of women's health focused through the lens of ovarian function. And fertility and menopause, big markers, but not the whole story. I've stopped calling them reproductive organs. So I think that calling them reproductive organs really
narrows our lens.
It narrows our lens. That's a good way to put it. Yeah, I think it forces you to pigeonhole all of women's health through the lens of fertility. And while fertility is very important, it's not the whole story. And I think it has allowed people to make women's health small. When it's not small, it's half the population, and it's not just about baby making.
So ovaries as these health-promoting organs.
The architects of health in female bodies, that's what I'm calling them.
Health of the female body, you heard it here. Because they are one of the first organs in the human body to see signs of aging, is there also potential to just better understand aging and bioactive peptides
Absolutely.
in all humans?
Yes.
Okay,
Yes.
I'd really love to drive home that point because
Yes.
I think that's what can get us all excited about this.
Okay, yes. There's several reasons to care about understanding ovarian aging, independent of whether you're female or you own a pair of ovaries or you have ever owned a pair of ovaries. There's the fact that reproductive span in females, so like the age at which a woman goes through menopause, so age at natural menopause, correlates with overall lifespan. So we don't understand that relationship. We don't know what's causative there. So it's just a correlation, but it's a strong one. And so women who go through menopause later tend to live longer. That does not mean that if you go through early menopause that you're going to die young. It's an average, it's statistics, it's a population, but it is a strong correlation. It's an interesting one. And I think if we could understand that relationship, it would give us a clue about what's driving ovarian aging, which is the key question we want to answer. But for men, brothers of women who go through menopause later also tend to live longer. So there's a genetic component there that's conserved. If we could understand what it was, would be important for aging in men as well. But then there's also just this idea that separately from female aging, the longevity space, the aging research space, which the Buck Institute is the first independent research institute to study aging, the whole reason we do that is not because we want to live forever, is because that period of time at the end of life when you've got multiple age-related diseases, and I'm talking about cancer, I'm talking about neurodegenerative disease, I'm talking about cardiovascular disease and autoimmune disease, all of these chronic diseases that we all experience in the modern world if we're lucky enough to live to old age. Your number one risk factor is your age. And so if that's true, then aging is kind of a public health pandemic because we're all living longer. And if my age is the number one risk factor for all those chronic diseases that are the major sort of economic burden on the healthcare system, then the older I get, the more likely I am to have one of or more of those things. The whole point of what we're doing is that if you can understand what's driving aging, like what are the mechanisms that actually cause your tissues to age, and you could intervene there, not to like reverse aging or any of this, that's not what we're talking about, but just to square off the curve essentially for healthspan, to exploit that knowledge to then be able to target all of those age-related diseases simultaneously. That's the goal, at least at the Buck Institute, to extend your health span. As we're making progress in that space, if we don't start to consider ovarian aging, then we're suddenly going to have women living more and more of their lives post-menopause, in this period of time when their health is compromised. And it is compromised. I'm sorry, I know people get upset when I say that, but the data is there. Like your risk for all of these chronic diseases goes up dramatically after menopause. It's important to say that ovaries are still doing something after menopause. They're just not doing what they were doing before.
Would it be fair to consider ovaries, and maybe I'm getting this completely wrong, but ovaries as kind of a perfect organ to study to better understand bioactive peptides in aging because ovaries are aging sooner. So studying what's changing and fluctuating may be a little bit easier because it's kind of ahead of the rest of the organs in our body.
That sounds like a really good story for how I got to where I am, but that's never how science works, right? We know so little about how they work and what's happening there that absolutely I want to characterize what's going on there in terms of bioactive peptides. I would actually even take a step back and say, you could think about ovaries as an accelerated model for human aging, which gives us a handle into studying aging that we have not had before. Because right now, if you want to test a longevity intervention, you're going to test it in a model system, a very imperfect model system. And you're never going to be able to do a real clinical trial in humans that measures lifespan or health span in old age because you would need to do it for decades. Right now, we test things in worms and flies and mice and rats and monkeys and dogs. Dogs are by far the best model system we have right now for mimicking human aging and testing interventions.
Better than monkeys.
Well, yeah. Non-human primates are maybe closer to us in terms of evolution, and there are some aspects of our physiology that are better modeled in a non-human primate, but Matt Kaberlein and others, Daniel Promislow, with the Dog Aging Project, have really pushed forward this idea that domesticated dogs, we accidentally built a really great model for aging in humans because what you have is a lot of different breeds of dog that are pretty much all the same species. But among that same species, you have this incredible diversity of size, shape, function, lifespan, behavioral tendencies, which diseases they're more likely to get. You have this real expansion of things that very much seems to mimic the inter-individual variability that you see between humans, right? They're different colors, they have different propensity to get cardiovascular disease. Some are big, some are small, some live a long time, some are more likely to get arthritis, like all these kinds of things. And they do tend to have a lot of the same age-related diseases. So it's a fascinating model in the Dog Aging Project. And there's also a company that's grown up around this called Loyal that's doing some really interesting stuff. But my point is, these are imperfect systems. That's the best that we've got right now, and I'm so glad that that's being developed. But hey, now we have this system that we can think about in young women. I mean, I think of you as being very young.
Thank you.
Young women where, you could imagine, well, actually, I used to say you could imagine. Somebody's actually doing this right now.
Oh, really?
But you can imagine testing an aging intervention where you do a clinical study and use ovarian endpoints as a proxy for aging in the rest of the body. And so you might imagine like a 2 or 3 year clinical study where you measure ovarian endpoints, not because you're trying to develop something to promote fertility, but rather
shorter study window.
Exactly, for something that's actually measuring aging in a human.
Cool.
So that's really cool. So ovaries are like the time machine we need to really explode the ability to test interventions in the longevity space.
I'm going to leave this conversation with so much appreciation for my ovaries. And so thinking kind of in the bigger picture, you said earlier that the receptors of bioactive peptides are very druggable. Mm-hmm. But I've also come to understand that there's different receptors, there's different bioactive peptides, and as you've mentioned previously, there's also enzymes that can degrade bioactive peptides. So there's so many points of intervention. Is there
regulation?
Regulation.
Yeah.
Okay. Is there one specific point that you think might be the most promising if we were to try to extend our health span all the way up until the end of our life?
No, but that's what's beautiful about it. The complexity is what makes it so exciting, because for different peptidergic systems, you might target different parts of the system depending on what you're trying to achieve, right? And so peptidases, those enzymes that break down peptides, they can specify both the path that a peptide can travel to signal and also the timing, right? And so it's actually, if you think about it, it's a beautiful way to shape the signal. Sure, you can dump peptide into a mouse brain. Like using a cannula, you can dump oxytocin into a mouse brain and elicit repeatable, robust behavioral phenotypes that have nothing to do with what the peptide does naturally. And that's because for signaling with peptides, it's important when, where, and how much. And so you can flood a system with peptide and get spurious results that have nothing to do with the natural functioning of the peptides. So that's why we do loss of function experiments where we're doing genetics manipulations and not gain of function, because gain of function gives you results that aren't really there. But it also gives you an appreciation for how important all of those different things are. And so what that means in practice is, like, if we just keep carrying forward this idea about oxytocin or vasopressin, there's a disease called diabetes insipidus. It's a little bit confusing because when I say diabetes, you probably think metabolism and insulin.
Yes.
Yeah, I don't know why they had to use the same word, but diabetes insipidus is not related to insulin signaling at all. It's a mutation actually in the vasopressin receptor 2, which is the primary kind of vasopressin receptor that you find in kidneys. And when you have this mutation as a human, it means that you have a really hard time regulating fluid balance. And so it comes with all kinds of really dramatic phenotypes. Anyway, you can treat diabetes insipidus by monkeying around with the receptor, but vasopressin receptor 2 is only expressed in the kidney. So if you're targeting vasopressin receptor 2, you're not going to hit the receptors that are expressed in the brain or the things that, mediate some of the social behavior. And so you can start to sort of tease apart once you understand how the system works, but you have to first understand how the system works. Does that make sense?
Yes, you're out on the frontier figuring it all out so that we can understand.
Not just me, there's lots of people who do this, but yes, this is where my passion lies.
But you do it too.
Yeah.
I have so many different questions I want to get to in this conversation, but I'm conscious of time. I first want to ask something that I would have been wanting an expert's opinion on for a while. So there's a growing number of people who are excited by the idea of living forever, and that if we can kind of tweak our physiology in the right way, that will be possible. As someone who studies bioactive peptides in aging, what are your thoughts on this proposition?
I get asked that question a lot. And I have strong opinions. Anyone who knows me knows that I'm very happy to share my opinions. I think that for me, the most pressing and also the most tractable question is how to extend health span. I think that there's a lot of value in moonshots, and I think there's a lot of value in debate. In a philosophical way, I love to talk to people about questions like, is there a limit to human lifespan? And is aging programmed? And in fact, those were debate topics that we engineered into a course that I developed or that I helped restart at the Marine Biology Lab, which is there to train the next generation of leaders in the field. Thinking through how to talk to these students and how to really instill in them a real ability to navigate all these kind of really high-level, complicated questions without just lecturing at them and telling them what I think. So I think that's really valuable. And I think in the aging space, we have what I would consider a fringe element that's full of people who just say they want to live forever, which is certainly one way to go. But I don't think there's any evidence that that's feasible for humans. And we can extend lifespan in many model organisms. We have done so for a long time, right? The first gene that controls aging was discovered in C. elegans, AGE1, which is, part of the mTOR and insulin signaling pathway. And if you think about that lifespan and you cut it into two parts, the part where you're healthy and the part at the end of your life where you're not healthy, when you extend lifespan in a model organism, so in flies and in worms and in yeast, in mice, in rats, monkeys, when you extend lifespan for the most part, those two things scale. And so to me, that's a nightmare scenario. So you live to be, however old you live to be, let's say we push it out to 200, but then the period of time when you're sick gets longer. That sounds terrible to me, and also would be economically infeasible, and there's a lot of reasons. So that's a very important piece of data to keep in mind. But the other thing is, like I said, I like dreamers, and I like these kind of moonshot ideas, but I think it really distracts from the real conversation, and it takes resources away from doing the science that would actually impact your life in a positive way. And we have literally no evidence that it would be possible. So that's kind of my take on it.
Beautiful answer. I mean, I personally would love to feel great at 90 rather than necessarily live to 100.
Yeah, that's right.
So great.
And that's doable. I feel like we've made a huge amount of progress in making that happen, and that's doable. The other thing I would say is that it is important, I think, one of the things that frustrates me about this field is experts, people who should know better, often will mix up truth and things that are supported by data with a bunch of nonsense that is unsupported by data, snake oil, if you will. And it's really impossible for somebody who's not a scientist and an expert in the space to untangle this nonsense.
Thank you for saying this, because I feel like this is the journey I'm constantly on. And it's one of the reasons I started this channel, because I'd like to start untangling the nonsense from the non-nonsense, because sometimes there are truly incredible strides in science that almost sound sci-fi, and there's a lot of data to back it up.
Yeah.
And so how are we supposed to know if something that's crazy sounds crazy just because it's novel?
Yeah.
Or because it's not substantiated?
Right. The media doesn't usually do a good job of filtering, and that's not their fault. Like, they're not experts. If someone who's an expert tells you something, you're going to believe it, right? For the most part. I think there is a huge vacuum and a big space for people like you who are hopefully going to translate some of this to the people who need to hear it. But I think some of the truth that's in those crazy things is that right now, lifestyle interventions, diet and exercise, and I'm talking about for normal aging, not for people who have actual disease that need to be treated, but for normal aging, I think diet and exercise, like how you move your body and what you put into it, are by far the best drugs. Like, by far, if you did a clinical trial, if you could do a clinical trial with those two things up against any drug, any intervention that we know about that changes the rate of aging or changes aging phenotypes in animals, they would win by a mile. And, hopefully we're making some progress in understanding, like, what is it about each one of those things that gives you that benefit, and that we can then, mimic those things. But we're not there yet. That's an important message. It really does matter how you move and what you put in your body. But that doesn't mean that you should be pumping yourself full of supplements.
Yes. This is amazing, because I think one of my personal approaches to health is also taking these daily non-invasive lifestyle interventions. These are things people rationally know they should be doing, but there's a behavioral and motivational aspect to it, which I wish we had more time to even start to touch upon the behavioral elements of bioactive peptides. I guess I'd love to know what your thoughts are on how there may be ways to modulate our behavior using any of these bioactive peptides, or is this also kind of a crazy idea and we should just keep striving to do things from our own willpower? But I'll caveat this question by saying I have noticed in my own lifetime, based on my physiology, that my motivation and behavior can change so drastically. So I'm wondering if there's any future in which we're almost able to make it a little easier to take these good steps for our health.
Yeah, you're talking about motivation, and certainly that's a whole area of cognitive science that is, tons of people work on that. I have a postdoc who's just finishing up her work, and she's literally going on the job market right now to become hopefully a professor somewhere and start her own lab. That is what she wants to work on. Cognitive circuits that control motivation, how they change with age, and how you might be able to intervene in the same way that I was talking about.
Much more eloquently put.
But this is important. And it is true that certainly your brain and neuromodulators, bioactive peptides are a big part of that, but lots of neuromodulators control your mood and can play a role in behaviors. And so the more we understand about how these systems work, the more there will be rational ways to try to promote that stuff. I will say GLP-1 is a neuropeptide, but the drugs that are, people are using now for diabetes and weight loss that are related to GLP-1 agonism, those drugs, they mimic what would be levels of peptide that would be at least 1,000-fold higher than what you would naturally have in your body. That's important to say for two reasons. One is it means we don't really understand how they work, because whatever they're doing, it's really not about the physiology of GLP-1 for the most part. And it also means that anybody who's claiming to feed you food or a supplement or something to increase your natural GLP-1, whatever they're able to do there, even if it's true, and it's probably not true, but even if it's true, is meaningless in the context of those drugs. So please don't take supplements that are that are telling you you're going to increase your GLP-1.
Are we going to look back on this time of this craze of GLP-1 agonists and think, what were we doing?
No, I don't think so.
Okay.
And this is not my area of expertise, but I think we're just at the beginning of unlocking an area of pharmacology that is profound, right? I mean, if you look at what's happening now, these drugs have been used in humans for a long time, like the precursors to these drugs, to treat diabetes. And so we have long-term safety data on those drugs. Usually when you have a new drug, the safety profile is a little bit like, you had the phase 1 to make sure it's not going to do something terrible, but the long-term safety profile is something that you discover as people take it long-term, but we have all this data that already exists. And of course, there are things that you have to be aware of and, lots of reasons why you would want to work with a doctor to make sure that everything is okay. But these are drugs that are allowing people to really change their physiology in dramatic ways, right? And none of them probably are perfect the way they are, but there's so much development going on that I think is very promising, I guess I would say. Not without caveats, but very promising. You could take someone who has struggled with weight, for example, for their entire lives. And obesity is not a choice. It's not something you can control. It's certainly not something you can do mind over matter and motivation to mitigate. It's a physiologic problem. Here's a class of drugs that seem to really help with that. So that's incredible, right? It's amazing.
Yes, it is amazing.
Yeah.
Well, you've already shared that, people can take care of what they put in their body and how they move their body. Are there any last words for someone watching who may be trying to do their best to extend their health span?
Yeah, I would say don't take supplements unless you need them. That's something that, again, is part of what plagues our fields, is that people think it's either neutral, like it's not going to do any harm, or it's positive, beneficial. But actually, supplements can do harm. And so just taking a bunch of things without really needing them is probably not a great idea, I would say. And I'm not a medical doctor. I'm just telling you what I see in the data.
We've just seen the tip of this iceberg.
Yeah, I'll keep talking. I can talk forever.
Thank you so much, Dr. Garrison. It's been a pleasure. I would love to know what you're taking away from this conversation. Do you have any other questions for Dr. Garrison that we might be able to address in a part 2? Thank you guys so much for watching, and I look forward to seeing you next time.